Welcome to this scientific exploration of how molecular properties define the taste of food. Today we're going to dive deep into the chemistry and biology of taste perception. Before we explore molecular properties, let's ask a fundamental question. What exactly is taste? Is it just flavor? Taste is actually a chemosensory perception. It's the detection of chemical compounds by specialized receptor cells on our tongue. Here's a critical distinction. Taste is not the same as flavor. Taste refers specifically to the 5 basic tastes we detect on our tongue. The 5 basic tastes are sweet, sour, salty, bitter, and umami. Each of these tastes is triggered by specific molecular properties. Here's a visualization of the 5 basic tastes. Each taste is mediated by different molecular interactions with taste receptors. To understand how molecules create taste, we need to understand taste receptors. These are protein structures embedded in taste bud cells. Each taste bud on your tongue contains 50 to 100 taste receptor cells. These cells contain specialized proteins that bind to taste molecules. Different taste receptors respond to different molecular properties. This is the key to understanding why certain molecules taste the way they do. Let's start with sweet taste. What molecular properties make something taste sweet? Sweet taste is detected by a heterodimeric receptor complex made of 2 proteins, T1R2 and T1R3. These proteins form a binding pocket. For a molecule to taste sweet, it needs specific structural features. First, it needs hydroxyl groups, which are OH groups that can form hydrogen bonds with the receptor. Second, it needs the right size and 3-dimensional shape to fit into the binding pocket. Think of it like a key fitting into a lock. Here's a simplified representation. The hydroxyl group on the left can form hydrogen bonds with amino acids in the receptor binding site on the right. Let's look at a concrete example. Both glucose and fructose are sugars with the same molecular formula, C6H12O6. Both have the molecular formula, C6H12O6. Yet fructose tastes about 1.7 times sweeter than glucose. Why? The answer lies in their 3-dimensional structure. Even though they have the same atoms, the spatial arrangement of their hydroxyl groups is different. The hydroxyl groups in fructose are positioned in a way that allows stronger and more numerous hydrogen bonds with the sweet receptor. This brings us to artificial sweeteners. How can we engineer molecules that are thousands of times sweeter than sugar? Sucralose, found in Splenda, is about 600 times sweeter than table sugar. The key modification is replacing 3 hydroxyl groups with chlorine atoms. This changes the binding affinity dramatically. The chlorine atoms increase hydrophobic interactions with certain regions of the receptor, making the binding much stronger and prolonged. Now let's explore sour taste. What makes something taste sour? Sour taste is fundamentally the detection of acidity. It's all about hydrogen ions, or protons. The more H+ ions, the more sour the taste. The mechanism involves specialized ion channels that allow protons to enter taste cells. This includes PKD2L1 and PKD1L3 proteins. Citric acid in lemons is a perfect example. When citric acid dissolves in saliva, it releases hydrogen ions. Citric acid dissociates into H+ ions and citrate anions. The H+ ions trigger the sour receptors. This is why the pH scale correlates with sourness. Lower pH means higher H+ concentration and stronger sour taste. Here's a pH scale showing where common sour foods fall. Lemons are around pH 2, which is highly acidic and very sour. Let's move to salty taste. This is perhaps the most straightforward taste from a molecular perspective. Salty taste primarily detects sodium ions, though other metal ions can also trigger it to a lesser degree. When table salt, sodium chloride, dissolves in saliva, it produces sodium ions. The mechanism involves epithelial sodium channels, abbreviated as ENaC. These channels allow sodium ions to directly enter taste cells. When sodium ions flow in, they cause the cell membrane to depolarize, triggering a neural signal to the brain. Here's an interesting question. Why do we crave salt? From an evolutionary perspective, sodium is essential for life but rare in nature. Sodium is crucial for nerve impulse transmission, muscle contraction, and maintaining proper fluid balance in our bodies. Our ancestors had very limited dietary sources of sodium, so we evolved a strong preference for salty taste to ensure adequate intake. Now let's examine bitter taste. This is the most complex and fascinating taste system. Humans have about 25 different bitter taste receptors, called T2R receptors. This is far more than any other taste. Why do we need so many bitter receptors? Because bitter taste evolved primarily as a defense mechanism against poisonous compounds. Many toxic compounds in plants are alkaloids, which are nitrogen-containing organic molecules that taste intensely bitter. Caffeine is a classic example. It's an alkaloid with a complex ring structure containing nitrogen atoms. Here's a simplified representation of caffeine's structure. The nitrogen atoms are key to its bitter taste and its interaction with bitter receptors. Interestingly, there's huge genetic variation in bitter taste sensitivity. Some people are super tasters, while others are non-tasters. The classic example is phenolphthaleocarbamide, or PTC, and propylthiouracil, or PROP. About 70% of people find these intensely bitter. But about 30% of people taste nothing at all. This is due to genetic variations in the T2R38 receptor gene. Finally, let's explore umami, the most recently recognized basic taste. What is umami? Umami means savory or delicious in Japanese. It was first scientifically described by Kikunae Ikeda in 1908. Umami primarily detects glutamate, which is an amino acid, and certain nucleotides, like inosine monophosphate. Glutamate has the molecular formula C5H9NO4. Umami is detected by the T1R1 and T1R3 receptor heterodimer. Notice that T1R3 is shared with sweet taste, but the partner protein is different. The key molecular feature for umami taste is the presence of carboxylate groups, which are negatively charged at physiological pH. Here's the carboxylate group that's essential for umami taste. The negative charge interacts with positively charged amino acids in the receptor. The most famous umami compound is MSG, or monosodium glutamate. It's simply the sodium salt of glutamic acid. Contrary to common misconceptions, MSG is naturally present in many foods like tomatoes, cheese, mushrooms, and meat. Here's an important question. Why do proteins taste more umami when cooked? When proteins are heated, they break down. Cooking breaks proteins into free amino acids, particularly glutamate. This is why grilled meat, roasted vegetables, and aged cheese are so savory. Let's discuss a broader principle. How does molecular size affect taste perception? Small molecules like sodium ions or simple sugars can easily access and bind to taste receptors. Very large molecules like complex proteins or polysaccharides are generally too big to fit into taste receptor binding sites. They taste bland. This is why starch, which is a polymer of glucose, doesn't taste sweet. The glucose units are locked in a large chain that can't bind to sweet receptors. But when you chew bread for a while, it starts to taste slightly sweet. Why? Because amylase enzyme in saliva breaks starch into smaller glucose units. Another crucial property is molecular hydrophobicity. How does water solubility affect taste? For a molecule to trigger taste, it must first dissolve in saliva, which is primarily water. This means it needs some degree of water solubility. Highly hydrophobic or water-hating molecules simply won't dissolve well enough to reach receptors in sufficient concentration. This is why pure fats and oils have minimal direct taste. They're highly hydrophobic and don't dissolve in saliva. However, recent research suggests there may be a specialized fat taste receptor called CD36, but this is still being actively studied. Now let's explore a fascinating aspect: stereochemistry. Can mirror image molecules taste different? Many organic molecules are chiral, meaning they have mirror image forms called enantiomers. These are like left and right hands. Because taste receptors are made of chiral amino acids, they can distinguish between enantiomers. This is called enantioselectivity. A classic example is carvone. R-carvone smells and tastes like spearmint, while S-carvone tastes like caraway. Same formula, different spatial arrangement. Aspartame, the artificial sweetener, only works as the specific stereoisomer that's manufactured. The mirror image has no sweet taste at all. Let's consider how temperature affects molecular taste perception. Does temperature matter? Temperature absolutely affects taste perception, and it does so through multiple mechanisms. Warm temperatures generally enhance sweetness perception. This is why ice cream tastes less sweet when frozen than when melted. Cold temperatures reduce bitter perception. This is why beer is often served cold. It masks bitter flavors. The mechanism involves temperature-dependent changes in receptor protein conformations. Temperature also affects how quickly molecules bind and unbind. An important phenomenon is synergy. Can molecules enhance each other's taste? First, there's mixture suppression. Some tastes can partially cancel each other out. Salt suppresses bitterness, which is why a pinch of salt improves coffee. Conversely, there's mixture enhancement. Glutamate and nucleotides show strong synergy in umami taste. When you combine glutamate with inosine monophosphate, or IMP, the umami intensity increases up to 8-fold. This is why dashi, which contains both, is so intensely savory. This synergy works because both molecules bind to the umami receptors simultaneously, creating a more stable and longer-lasting activation. Let's discuss practical applications. How do food scientists use this molecular knowledge? Food scientists design flavor enhancers by understanding receptor molecule interactions. They can create compounds that amplify specific tastes. For health reasons, reducing salt while maintaining taste is crucial. Scientists develop salt substitutes that trigger sodium channels without using sodium chloride. Many medicines are bitter. Pharmaceutical companies use bitter blockers, molecules that competitively bind to bitter receptors without activating them. To reduce sugar content, high-intensity sweeteners are designed. These bind more strongly to sweet receptors, requiring much smaller quantities. Let's summarize the key molecular properties that define taste perception. First, molecular structure and three-dimensional shape determine whether and how strongly a molecule binds to taste receptors. Second, specific functional groups like hydroxyl, carboxyl, or amino groups enable hydrogen bonding and electrostatic interactions. Third, molecular size must be appropriate. Too large and it won't fit. Too small and it may not bind strongly enough. Fourth, sufficient hydrophilicity is needed for dissolution in saliva and transport to receptors. Fifth, stereochemistry matters immensely. Mirror image molecules can taste completely different. Sixth, the ionization state of molecules, which depends on pH, affects their binding to receptors. In conclusion, taste is fundamentally a molecular recognition phenomenon. Each taste is the result of specific chemical structures interacting with specific protein receptors. Understanding these principles allows us to rationally design flavors, improve nutrition, and create better-tasting medicines. Future directions include personalized nutrition based on individual genetic variations in taste receptors and development of novel taste modulators. Thank you for joining me on this molecular journey through the science of taste. The next time you eat, remember the incredible molecular ballet happening on your tongue.